You have just been chairing a riveting session here in ESMO about a really cutting-edge issue, that's molecular profiling. Full of promise perhaps that isn't as fulfilled as we would like. Can I take you through some of the things? First of all, let me ask you, what for you was the most exciting thing about the session you've just been chairing?
What we tried is to come to an actual positioning where we are with regard to personalizing medicine, and we invited persons with different knowledge contributing to that. And the summary, if I do it very shortly, I would say we came to the conclusion that we are approaching personalized medicine, but we are still on the way from a stratified medicine to a personalized medicine. We haven't reached that goal yet.
Everybody wants to have personalized medicine. Everybody's talking about personalized medicine, but everybody wants the drug that works, and often that's the same drug for many different people.
I mean, this is an eternal wish that we want to personalize medicine, and we already wished that ages before, and at that time we used different methodologies to assess whether treatment according to a specific test system was better than the regular treatment. And so far, none of the systems has provided us with such convincing results that we can say we have proved it, but we never have been as far advanced as we are right now.
Okay, there are tantalizing data coming out about the identity of certain molecules. There was a talk by Dr. Mandrekar about designing trials which would actually, hopefully, enable you to use some of that molecular profiling knowledge. What came out of that?
It is key to use the right methodology to get the right answer. And a specific topic Sumitra Mandrekar was raising was the issue of patient enrichment. If we are dealing with so-called targeted therapies, it's clear that we want to have those patients in the trials who have those targets in their tumors. And the crucial question, which is still difficult to answer, is when is the best situation to enrich with these patients, or when can or have we still to do trials without enrichment. So with the developmental I would say at the late phase 1 stage or at the early phase 2 stage, it's time to enrich in case there is a low penetrance of that phenomenon. Otherwise, one would not find out the efficacy of drugs directed against such targets.
What you're saying is that some features are at a low numerical value, a low number of patients who have certain molecular features, but they might collectively have a big impact on cancer.
Right. And if we already know that the phenomenon is rare, we have to envisage that, and we have to adapt the trial methodology. We would never have detected the potency of trastuzumab if we would not have enriched this patient early on, and we would not have done adjuvant trials which demonstrate now already survival benefit.
Okay, there are already trials that are testing profiling, the efficacy of profiling in clinical trials. Jean-Charles Soria gave an overview. What emerged from that?
He focused on lung cancer. This was also the tumor entity where that topic started with the so-called battle trial in the MD Anderson, but in Spain, years before already there was a trial from the group of Rafael Rosell, who tried for the first time in the world to compare in a randomized fashion whether treatment according to genomic profiling was better than regular routine treatment. Unfortunately, this first European trial did not meet the endpoint, and it took a while until take another endeavor, and the battle trial was a more sophisticated trial and was successful. And now there are even much more sophisticated trials ongoing, randomized trials, really comparing treatment according to the molecular profiling versus treatment according to the best control. But all of these trials are still ongoing, and they are from salvage treatment or remission maintenance in the first line until adjuvant setting, but none of them has any result yet.
Now, the enemies of all this are things like tumour heterogeneity and also the fact that you can have targets, you may have many targets, but you can't necessarily, they can actually develop or change during the life of the cancer, so to speak. So how do you cope with that? Is it not too complex?
It is very complex. One way of coping with it is to have a permanent surveillance of the actual molecular situation of the patient, to do so-called longitudinal survey, to do rebiopsies, which would be easier to do via liquid biopsies, either by circulating tumor DNA or by testing circulating tumor cells, to get the actual heterogeneity of the tumor represented and to be able to react pharmacologically. Another approach is to combine drugs to avoid the early occurrence of resistance, to produce drugs specifically conceived for those patients who already got a resistance, but it's an imminent phenomenon, and maybe at the end it's not always to overcome.
Of course, you kind of summed up some of the philosophy of all of this, quoting a famous poet saying, "Targets, targets everywhere, nor any drug to treat." It's difficult to find a drug to actually treat some of these many targets. Do you see any hope in the future?
Indeed, it is a difficulty, but there is no alternative to be optimistic into my eyes. And there are also already ways to do so from the point of view of what's available to us from a chemical point of view. Many more compounds could be defined which are not yet there to target, and the so-called undruggable targets also could be druggable in the future, directly or indirectly. And one way of overcoming situations of resistance may be permanent longitudinal assessing of the actual or monitoring of the actual genetic situation, and to combine drugs accordingly to these patients.
Right, there's a lot of work to be done there scientifically. What should cancer clinicians make of these data now? What's the take-home message?
To be optimistic, but at the same time to stay realistic, to use those compounds which are out there, not to trust in announcements which may be commercially based, already to use tests available in some countries right now at high prices without sufficient background from the methodology.
Do you foresee an era when targeting 2 or 3 different molecular features could make a big difference to cancer treatment?
Yes, I do indeed. I see that times coming. In some instances, in some trials, we already got positive feedback, but this is still, let's say, in the experimental situation, and it's not in the clinical routine yet.